name is Margarida Carris, and I am the project manager. We're going to start this session by briefly addressing the two entities behind this project. I will now pass the floor to Carolina Guedes, CEO of Lisbon PH.
Hello everyone, my name is Carolina Guedes and I am the current CEO of Lisbon PH, and I'm here to pronounce to you Lisbon PH, the Junior Enterprise of Faculty of Pharmacy of the University of Lisbon. We are involved in a junior enterprise movement that is a non-profit, non-political and non-religious organization only managed by university students who are the first to join the youth to the entrepreneurship. We are all around Europe within more than 370 junior enterprises, and in Portugal we have 25 that is distributed to 17 educational institutes. Regarding Lisbon PH, we were the first federated junior enterprise on the healthcare sector. We are also a non-profit organization founded and managed by a group of entrepreneurial and dynamic university students who are eager to add value to their base education. We develop projects focused on accessible training for all healthcare professionals. We are guided through a clear vision for the development of the health professional of the future, entrepreneurial, creative, and multidisciplinary, and guided by 7 core values. We have developed so far more than 300 projects within our 10-year of history, based in our 3 core business. The first one, events, where we organize, manage, and promote events on relevant healthcare topics, whether in person or in online, like these webinars. E-learning courses are online interactive courses that are accredited by the National Society of Pharmacists, and our consulting and logistics, where we provide consultancy service to your enterprise. We have trained so far more than 45,000 healthcare professionals. Webinars is a major platform that we have been exploring since the COVID times. So if you have a project that you want to share with the community, we are now focusing so much on the research, and we want to help other enterprises to release their new information. So if you want, you can contact Lisbon PH and we can collaborate in the future. We also have some e-learning courses available for you to do right now. We want to present to you Job Ready, our international e-learning course, Get Ready for Your Future. It's a €10 e-learning course that is available with 12 hours of training. Regarding our consultancy services, we want to bring more innovation, more quality, more flexibility with a competitive price to your enterprise as well. Lisbon PH is also available in an audio platform. Out of the Health Box is the name of our podcast. We monthly launch 2 episodes based on topics that are relevant in the healthcare sector, but also in the entrepreneurial ones. And we are available in all the audio platforms, Google Podcasts, Apple Podcasts, and Spotify. Lisbon PH is also present in many social networks, Facebook, LinkedIn, Instagram, and YouTube, where globally we can count with more than 19,000 followers, and our mailing list with a lot of contacts as well, mostly healthcare professionals, but students make part as well. Sustainability is one of our main core values, and so we have our PH Plus project and program that is our sustainability and corporate social responsibility program. Is focused on 3 main pillars: health education, solidarity causes, and ambiental responsibility. We have so far been able to impact 16 out of the 17 SDGs established by the United Nations. And if you want to receive exclusive offers, follow closely our work and be the first to enjoy our projects, make sure to scan this QR code so you can be in contact with our newsletter and receive all of our information. Thank you so much and have a great webinar.
Thank you very much, Carolina. Next, I pass the floor to Dr. Ana Cadet, founder of the Non-Conformist Scientist.
Thank you, Margarida, and hello, everyone. Good afternoon and good night, and welcome to the Health Innovation webinar that we are presenting today. It's my great pleasure to introduce you to the Non-Conformist Scientist, known as the NC Scientist on social media. So if you haven't done so, go to Instagram and start following this platform. And also, if you want to get in touch to what's going on in our project, just scan the QR code. I also added it into the next slides, so you can subscribe to our newsletter and don't miss the programs that we are going to offer. My name is Ana Cadet. I'm a PhD. I'm the founder and president of the Nonconformist Scientist, and you can find more about myself on LinkedIn. So, welcome to the Nonconformist Scientist. We are a non-profit organization based in Boston, in the US, with a local chapter in Portugal. Our team is composed of Portuguese women working around the globe, united by a vision to empower scientists, no matter where they live, to perform at their best. To do so, we are offering 3 different projects. She Has a PhD, it's our podcast. It's currently available on Spotify. You can watch the episodes on Apple or any other podcast platform that you use. Our major goal is to inspire the new generation of female scientists to become leaders. If you see, there's only a small percentage of women in leadership positions. When it goes to women in scientists, it's even less. So we want to break the rules and show to more girls in science that if that's the pathway you wanted to take, there's stories of other women, and we want to tell them, we want to show you the way. So one day, hopefully, if you dream to, you can be a leader as well. We organize webinars. An example is the webinar we are organizing in collaboration with Lisbon PH. So our events tend to be for global education. We believe that by the privilege that we have by working, in this case in Boston, or in the best universities and companies, that we can pull together extraordinary people to then provide and share their knowledge with all the scientists globally. So we have webinars where we share knowledge, and we also have webinars dedicated for the career of scientists. In this space, we also have our career hub, and we work in partnership with companies and institutions. And the major goal here is to advance the professional and personal life of scientists so they have the tools they need to progress in their careers. If you want to learn more about what we are doing and are interested in the career that a scientist can take, we invite you to participate in our next webinar. It's going to be held on next Tuesday. It's the Career Path in Science webinar. It's going to be on May 21st, and we are going to have a person from academia, another one from industry, and a coach that is going to help students transitioning from science, from an academia pathway into the industry. So just go on Instagram to NC Scientists. You are going to see the publication that I have. We share these on our newsletter. We also use LinkedIn. We are building the platform. It's not like Lisbon PH is way ahead of us. We are still starting. But if you're interested in this type of webinars, just start following us and you'll get to see the news. I cannot finish this presentation without acknowledging the team. For the events team, we have these impressive women working with us. And if you're interested in collaborating with us, if you have ideas, if you have suggestions, just send an email to events@ncscientist.com and we'll be happy to talk to you. So thank you so much. Our next event is the Career Path in Science. But for now, I hope you enjoyed the Health Innovation webinar and what our experts have to say in artificial intelligence for drug discovery. Thank you so much.
Thank you, Dr. Anna. After introducing the 2 entities that have made this series of 3 webinars possible, I'm pleased to present the theme of our 3rd and last webinar, Artificial Intelligence in Drug Discovery. Introducing this topic, we have Dr. Rani Powers, founder and CEO at Pluto, and Dr. Christina Ulikna, research scientist at Valence Labs. Thank you so much for accepting our invitation and sharing your knowledge. Additionally, I invite the audience to raise any questions they would like to see addressed on this topic in the chat. These questions will be presented after exploring the theme. I now yield the floor to each of the speakers, and thank you once again.
Thank you so much, Margarita and others, for the opportunity and invitation to speak with the group today, especially one organized by such an impressive group of women. My journey in life sciences is something that I'll touch briefly on today during the presentation, but I just wanted to say thank you, and it's really inspiring to see the work being done by your organization. So we're really focused on innovation today. It seems like this will be a great theme for the talk. And I think especially over the past year, we have all been inundated with the amount of innovation happening in AI, so much so that it's hard to keep up with all of the new papers being published. So rather than kind of regurgitating all of this, innovation or really drilling down into specific areas of data, I really wanted to share something a little bit different. So although the number of computational folks in life sciences is definitely increasing, and we know that, there is still an estimated order of magnitude minimum difference in the number of computational life sciences versus what we think of as more traditional life sciences researchers. And so I wanted to focus my talk today on practical considerations and exciting developments in the application of AI. Practically speaking, how can you use AI when you're doing smaller scale studies? And I put smaller kind of in parentheses here because I kept looking for a different word to describe these studies. I think smaller is the most accurate, but we'll kind of get into what I mean by that. I did put omics in here, and I'll speak a little bit about the kinds of data types that I've worked mostly with in my career. But hopefully these kinds of thought-provoking principles you can apply regardless of the type of data that you're working with. And so really the point of this talk is hopefully to inspire some new ideas, especially for the folks really focused in life sciences and biology that are on the call today, maybe even students or postdocs working on their research, and how you can think about applying AI. Great. So I think everybody will agree data sets are becoming absolutely massive when it comes to AI in drug discovery. So you may have heard the phrase lab in the loop. This is a really fantastic concept being talked about in various aspects, especially in big pharma. Dr. Aviv Regev spoke about this in her keynote at AACR last month also. And so I'm showing here kind of this lab in the loop diagram, really pioneered by NVIDIA and Genentech. And then on the right side, Dr. Aviv Regev at her AACR talk. If you were there at AACR, you might have even heard some of the murmurs in the crowd when Dr. Regev was talking about if the data is large enough, it can actually outweigh other measures of quality in some respects. But overall, this lab in the loop concept really refers to a lab generating data, which can be aggregated in very large quantities and then fed into an AI model, which has to be trained with, again, a very extremely large data set to ultimately get to groundbreaking insight in biology, specifically where I'm focusing. And so the concept of using these massive data sets to train models and derive that groundbreaking insight is a really wonderful story that's being told at many, many conferences nowadays. These stories are surely going to be the whales that contribute to breakthroughs in many different areas of science. Something that I think is really incredible here is that computational biology and AI specifically, is everywhere. We used to, 10 years ago, we had to go to a specific computational biology focused conference to really hear about AI. But this is at AACR, one of the largest cancer-focused meetings in the US, and AI really took center stage here through the keynotes. So the concept, is really incredible of using these kind of large scale data sets. But I wanted to pose the question to everybody, is every company that's doing drug discovery generating data sets at this scale? I think we think surely, while there's no data showing the amount of data generated by every pharma and every biotech company, anecdotally, we all know the answer to be no. At my company, Pluto, for example, we're proud to work with a number of biopharma companies whose scientists might only be looking at one next generation sequencing study per month. Now that we've been around for a few years, I've even gotten to see, for example, one experiment be the initial foundation for a drug being used in a new indication. And that was really based on the research done, from a single study. So today, as a former bench scientist, I wanted to talk about a different kind of story that we might not be thinking about as much when it comes to AI and drug discovery, which are kind of those micro stories. Those hundreds of thousands of biologists around the world are creating these scientific stories, potentially starting with small data sets. And many of those scientific stories will have the potential to make an impact in the field, whether it's transformative or important in an incremental way. those scientific stories that can start really with just 50 patient samples or even an N equals 12 mouse experiment. So I wanted to give a little bit of context into why I started thinking about these problems. And then kind of second question here, are we required to generate massive data to leverage the benefits of AI? So hopefully I've explained, no, I don't think that's the case. So some quick background here. As far as my timeline in computational biology, science has always been a passion of mine. But I actually worked early on in my career as a video game developer. So I've worked in software completely outside of life sciences. And this is really where things started to collide for me, where I saw these beautiful user experiences being shown to users, to a large number of users in, for example, the video game industry, but wasn't seeing that same level of user engagement or user experience being applied to science. I did my PhD in computational biology to bring these things together, but I was still working in software startups developing consumer-facing or user-facing applications. And in these kinds of applications, the way that the human is focused on the design or interacting with the design of your application is very important for them actually achieving success with your product. So initially I started in a product, which was a video game, but started to move later in my career towards products that were life sciences focused. And interestingly, in the genomics field, for example, communicating complex information like your ancestry or genomics is actually pretty difficult to do from a user-facing perspective. These are, lay people that may not have the life sciences background, and so we use design and user interface to communicate complex scientific concepts. And all of this, I think, ties really well into where we're headed with AI and drug discovery, which I'll get to kind of in the subsequent steps here. Following my computational biology PhD, I went to the Wyss Institute at Harvard University, started to work more in a predictive and AI-focused role at the Wyss Institute, and eventually still was seeing this gap between the user experience that we had around allowing biologists to implement their own AI or ML or statistics or any kind of analysis, really. Versus the algorithms that we had available. And so this is what led me to found my company, Pluto Bio. So really what this kind of came down to across the journey that I had, that I have had so far in my career was just this real strong notion that we need to keep the biologist in the loop. So in addition to having our lab in the loop, to generating data, putting that into AI models, getting insight from there, it's really important that we keep the biologists, who are humans, in the loop. And so this is something that I wanted to focus on today and kind of use to inspire you all to think about how you, the biologists and life sciences researchers and scientists, no matter where you are in your career, really involved when we're thinking about AI as a potential co-pilot. So AI is particularly well suited to help with automating repetitive tasks or very large tasks, like the really large data sets that I was mentioning are now being generated, especially in cancer research. But our biologists, who are humans, of course, aren't going away anytime soon. And we really want to think about how we can leverage the biologist's knowledge in addition to having this AI co-pilot, this kind of superpower, when we're thinking about doing drug discovery. So I really believe that this co-pilot approach is going to help augment the interdisciplinary teams that we've already started building to do drug discovery. I think one interesting framework for thinking about this, for example, is the 5R framework, which I'm showing here, which was really pioneered by AstraZeneca. I think this was in 2018, really, that they had published this initially, and they've since been able to show how thinking about the right target, the right tissue, the right patient has made a real impact in their drug discovery. So I'd like everybody to consider similar kinds of benefits when we're thinking about the application of AI and how the explainability of a result is really beneficial for us actually in moving a drug through the pipeline. And similar to how we might have, rational drug design, we need rational experiment design still. So when we're thinking about designing our studies, as small as they might be, AI can actually play a part in helping us with that as well. So another reason, kind of practical consideration here, and a reason that I wanted to talk about applications of AI to small data sets, are there very legitimate reasons for having small data sets. In some cases, we won't ever be able to generate the scale of data that other companies are generating for particular indications. It wouldn't be a talk based on omics if I didn't show this pretty famous graph here to show that the cost of sequencing, for example, has come down dramatically over the past 10, 20 years. However, that cost, and I'm using next generation sequencing as an example here, that cost is still not zero. So we're averaging around $100 to $1,000 per sample for a next generation sequencing experiment. And that means that labs are still having to write grants, or biotech companies are still having to make a decision about which samples are getting sequenced and how they're designing those studies. So again, not every company can generate massive data sets that they're going to use to train their own AI models. And similarly, you may have populations that are rare in terms of the number of patients or difficult to recruit for other reasons. And then also just in terms of sample size, not having things like an immortalized cell line or a scalable model available to you can really mean that your sample size and therefore your data size is limited. So I've been talking a little bit here, back and forth around sequencing-based data. I wanted to highlight omics data in particular, in case this is useful to anybody. Omics data is especially applicable to AI, and AI can be well applied to omics data. So I wanted to put up a couple slides that just describe, the state of where we are with omics data analysis and different types. So these are kind of 4 canonical buckets that we think of when we're talking about omics data. And what we're doing with omics is measuring different kinds of molecules. So DNA, RNA, the binding of DNA and proteins, or proteins and metabolites. And so we have a bunch of different assays that we can use to do this. Omics data is really good for applying AI because the omics data types follow the same rough model. Even if you're applying those different assays that I was showing on the last slide, we'll always have sample labels. So any kind of observation that we're making, the sample will have a label. We know if that sample came from a healthy or a diseased patient or other populations. And the omics data comes in typically a raw format when you're performing the assay, but we can quickly transform that raw assay data into a tabular processed version. So when we talk about omics data and its use for AI here, what we're able to generate is very deep data where we might have tens of thousands of data points from each sample, and we know what that sample came from. So I wanted to give that brief intro there into omics data before we really launch into some of what I think are fun and practical examples in AI. So initially with omics data, we were using more traditional statistics or machine learning to analyze omics data sets. And again, this can really be applied to other kinds of biological data, but omics is kind of where we've specialized, where I've specialized, and I think it provides an interesting use case. So on the left is a really pretty basic statistical model that fits the assumption or the distribution of values that you would expect in this case, an RNA sequencing data set. This has historically been used in different ways for drug discovery, like target identification, meaning let's look at the top targets that have increased or decreased in expression in this experiment. And that, again, used really basic statistical models like a negative binomial distribution in this case. And then as machine learning has become more and more popular in life sciences, we can do drug discovery in even kind of deeper ways where, for example, we're classifying specific cell types and we're really honing in on which of the cells are we treating, or which of the cells do we want to be treating, or where is my target expressed in a heterogeneous tissue. And so machine learning here has been applied, and that could include clustering algorithms. We're all probably very familiar with dimensionality reduction. Also, this is a UMAP plot that's being shown here. And now we see the advent of AI being applied to these same kinds of data sets. So I wanted to highlight a really kind of fun example that we've been using for a while, which was applying ChatGPT to cell type annotation. So I was showing previously a UMAP plot that has different cell types here lit up as different colored clusters on the plot. Interestingly, what we had found pretty early on is that large language models are able to take in the gene expression data in this, very biological use case here, and rationalize what cell this particular cluster was being colored by based on its gene expression. And so I wanted to show a ChatGPT output here where in our product, for example, you're able to say, help me annotate this cell type. And ChatGPT, just plain ChatGPT, can provide you with, a top list of predicted cell types with rationale for why it had selected those. And you can really go as deep into this as you want. And it was interesting because when we had first started playing with this, we thought, well, surely, people are going to train better models and, we'll see how these will all end up performing. And sure enough, single cell GPT is now in bioRxiv. And so there has been some work published here, but the benchmarking is pretty incredible, just showing how large language models are able to do this, not specialized large language models, but very generalized ones. So that's, I think, a really interesting application here that you can use to, play with your own data and using these kind of commercial LLMs that are available without needing a very large data set yourself. Another fun example that we're using at Pluto right now in the context of just that single experiment is when the AI has full context of your experimental design, it can actually help you interpret what you're seeing. And so this gets a little bit meta if you're using an AI algorithm to generate a particular graphical output and then using AI to further interpret what that algorithm is producing for you. But this is shown in our product here and some LLM output examples where the AI can also suggest follow-up analyses to the researcher. So I wanted to end with, a couple of just, like I said, inspirational or hopefully thought-provoking ideas for ways that you can apply AI to your smaller omics data sets and really leverage the number of data points that you're gathering on every single sample with different, AI applications. So the goal here is really, or the metric when we're applying AI, I think, to smaller studies is to try to improve the amount of insight that we can gain from a single experiment or per unit of experimental data, like per sample, by using AI to really enrich. So now instead of just saying we've got 30,000 genes that we've measured per sample and we have some standard analyses that we can do, what else can we as biologists co-piloting, AI, are we able to ask from a deeper perspective? I did want to highlight also that it's very important to make sure that your data is okay to use. So if you're looking at one of these commercially available LLMs or even, your company's own models, it's really important to ask, is the data that I'm putting in here okay? How is this going to be used? Will it be used in additional aggregate training and so on? But really high level, just some inspiration that you can hopefully take from this talk and apply to your own experimental data. I showed a cell type annotation example. Literature search, companies like Scite.ai are taking, for example, the entirety of PubMed and other data sources and letting you perform literature search as assisted by AI. Now, this is really important for just even small scale studies because to enrich that amount of insight that we can get from a single data set, you can imagine that drawing on the wealth of literature that's available to understand within the context of your experiment, more of your targets and genes and pulling in citations can be incredibly powerful and also save you a lot of time. So while this is one that's getting a lot of buzz and, rightly so, I do think it's worth pursuing and trying out for your own experiments. AI can also be used and is heavily used now in just analysis or code debugging. So this is a great way to, again, deeper explore those small data sets that you have. And I'll say, in addition to really improving the amount of insight we're gaining per unit of experimental data, what we're trying to do with AI is expand the capacity without needing to expand headcount necessarily at your company. And so using things like literature search or code debugging aspects of large language models is a great way to expand that capacity. Additionally, cross-dataset comparison or even multiomics is an area that's under active development right now for AI in drug discovery. This is definitely something worth looking into, and I have lots and lots of references that would probably be too many to include here, but I'd be happy to share after the call for people that are, I think, doing this in new and innovative ways. Chatting with a large language model is not the most intuitive interface for humans, for biologists. However, chatting with a large language model is a great way to generate ideas for follow-up experiments. So that's something else. And then I really just, wanted to open up the floor. I'd be really excited to hear what else is your company doing right now to leverage small data sets, small studies, and just really kind of get us all thinking about this. I know we're going to hear, fantastic talks, continue to hear fantastic talks about large-scale data analysis and model training. But I wanted to put a plug in here for, the biologists that might be sitting with their smaller studies right now, thinking, how do I really start to use AI in practice, or even start thinking about, and gaining the confidence to do so. So hopefully time-wise that worked out all right. I'll end there and say thank you.
I thank Dr. Hani for enlightening us on this theme. The topics covered were indeed not only interesting, but also really important to address. I now pass the floor to Dr. Christina Ulikna.
Thank you very much for having me. Thank you so much, Margarita, for the introduction, and thank you for the opportunity to talk to all of you tonight. My name is Christina, as was already told. I am joining you here today from as a representative from Valence Labs, which are covered by Recursion. And I wanted to spend a little bit of my time at the beginning just kind of introducing my background and showing you how a biologist like myself can transition into the field of computational biology and join one of the most exciting AI research labs in the world and in the academic environment. So yeah, I am going to move on to the next slide, which is wrapping up my very early research journey that brought me to the point where I joined Recursion and Valence. So let me just walk you through what I did and what my kind of academic story has been so far. So about 10 years ago, I was very passionate about doing science. I was always a very good student in biology and chemistry at high school, and as it always is, a lot of people were encouraging me to do medicine for obvious reasons, but I never really wanted to do that. I was always interested in the kind of smaller scale analysis of cells and how we can understand what the molecules and all the complexes inside a cell work, and how we can understand and shine some light onto the processes that are so fundamental for our life. They actually bring us to either a healthy state or, unfortunately, sometimes in a disease state. And if we understand it, how can we actually leverage that knowledge to be able to correct it and to bring the person or the patient back to the healthy state? So trying to kind of balance the two a little bit, trying to stay in the interface of medicine, but also the encouraging myself to experience the scientific life. I decided to join baccalaureate course in biomedical science at King's College London, where I moved, and I had a fantastic time during my undergrad. I must say, I really enjoyed it, and I was really excited about learning about all of the cells, all about the research, and learning all about what it really means to be a scientist, what it actually means to do research, what it actually means to be in the lab. And I must say that during my time as an undergraduate in London, I was very fortunate because I had the privilege to join some of the most famous labs all around the world to do a couple of internships. So at the beginning, after my first year, I was super fortunate to join MIT at the Whitehead Institute, where I learned a little bit more about cancer in one of the most famous cancer biology labs in the world, where I learned about cancer specifically, but I also learned about what it would be like if I continued, maybe towards a research career, what it would be like if I continued towards a PhD, and what it would be like if I actually wanted to do innovative research. And that was a fantastic experience. I really loved it. I came back to my second year of undergrad fully motivated and wanted to seek for another internship for next summer. So then I joined the Cancer Research UK because, now after like 1 3-month internship, I was almost an expert in cancer, I thought. And so I really wanted to continue kind of thriving in this environment, learning a little bit more about science, learning a little bit more about research, and still remaining a solid biologist. I was spending most of my time at that time in the lab, pipetting, doing cell culture, Western blots, and so on and so on. And I really enjoyed it. After that, I actually thought that, well, the work in the lab was making me so happy that why not just interrupt my studies for a little bit and just join a company for about a year. My program at King's actually allowed this, so I could interrupt my studies, and I could actually, go for a little longer than just a 3-month internship. I could actually go and experience what it would be like if I joined a company more like on full-time or temporarily full-time matter. So that's what I did. I joined a smaller, at that time, relatively small company. In the meantime, they actually grew to a very successful place, and I joined them as a molecular scientist. I was doing a lot of pipetting, still the same techniques as I learned in my previous internships and in my undergrad. But towards the end of my undergrad and towards the end of that particular placement, I actually figured out, and I could really see that being able to automate some of the processes that the biologists were doing by hand was becoming more and more important. I saw that some of the procedures that the biologists were performing by hand at the beginning of that year actually were replaced by robots, and they were replaced by people who were able to analyze the data computationally, by people who were able to quantify the data computationally. And frankly, there was a lot of emphasis into getting a little bit more of the people who could work with a computer. And at the time, that sounded like a fantastic challenge. At that time, I must say that I wasn't even able to switch a computer on. I was at that stage because I was kind of believing that biology and the pipetting is all that I need for my success as a scientist. But towards the end of my undergrad, I was really becoming more and more convinced that it would be great to at least experience, at least to try a little bit of a new avenue. So what I did then was that I was, again, very privileged, and thanks to all of my internship experience, I was admitted into a PhD program. And my PhD was in a London interdisciplinary doctoral consortium called LIDO, and I really enjoyed that. The fact that in our first year, what they let us do is, instead of kind of committing to a certain lab, and instead of kind of just jumping straight into the PhD research, they let us do rotations. So the rotations effectively are almost like an internship, if you're familiar with that sort of model. And the rotations are really kind of trying to help you understand what it would be like if you joined that particular lab. And so you can not just experience what it would be like to work with those particular people in that lab, but also you can experience what it would be like to do and continue research on that particular topic. And because I was really, triggered, I really wanted to kind of explore this computational world, and I wanted to learn something new. What I did was that in my first rotation, I chose a very computationally heavy project. The program themselves, they were pushing us toward computation because they were already realizing the importance of it in the world. And so what I did was that for my first rotation, I was very transparent with my potential supervisor. I approached them saying like, hey, I know nothing. I can't even code a for loop. Could you please admit me to the lab because I'm very keen to learn? And frankly, that rotation didn't end up being the one that I chose to do, but I fell in love with the computational approaches so much, and I fell in love with the coding so much that I actually wanted to pursue the computational aspects of my research a little bit farther. So for my second internship, I committed to my real PhD my real PhD project, and I continued in this program up until about last year. So during my PhD, I was becoming, a little bit more acquainted to computational biology, and I was very happy that I was able to use all of the coding and the AI skills and the early segmentation masks and the development of the AI models using on image analysis data. But I still kind of lacked a little bit of the confidence. I didn't consider myself to be a coder because I still came from this biological background, and that really changed when I managed to score an internship in Microsoft Research, which was one of the, a really kind of like a turning point for me, kind of building my confidence. That's one thing, but also building the confidence of others that I can do this, and that biologists are also people who can learn coding and they can reliably analyze the data that they're given, so that they can train models that can help them understand all the data. After my PhD, I continued very briefly into my postdoctoral research associate position at the Alan Turing Institute, not very far away from here. It's based in London. And during my postdoc, I kind of naturally continued working on my PhD topics and on my PhD thesis to finish some of the papers up. But I was very kind of trying to I very early realized that perhaps postdoctoral position is not something for me. I was really kind of jealous of all the industry and all the the company giants that were able to handle large quantities of data. In fact, the week when I submitted my PhD thesis, the ChatGPT model, the first one, was released, and I was really puzzled by the amount of data that people can handle. And I really kind of was jealous of all the companies that had the availability of all the data that they were generating. So after my PhD, after my postdoc, I decided to do a switch from academia into industry. And I was actually very interested in joining a lab which does AI for drug discovery, which is would still remain my passion, but I really wanted to do it in a company where data is not even a question. It's where in a company where the question really is, how do we handle and analyze the amount of data that we have available? And this is what led me to the decision to join Valence Labs earlier this year. Hopefully, kind of like throughout my trajectory, I hopefully managed to explain that I was always very interested in the process of drug discovery, the process of understanding how they act on a molecular and chemical and cellular level. But hopefully, what I really want to introduce in the rest of my talk is how I'm interested in building biologically meaningful AI models, so that we can not just use AI just for the sake of saying that we're using AI, which is what I feel like a lot of people in the field are inclining towards, but to use AI to make sure that we're using it for discovery, whether it's drug discovery, that's one aspect of it, but whether it's the biological discovery and the discovery of the processes that we're actually initially interested in, that's really what I want to use AI for. So let me just walk you through how I got to the project in Valence. So I just very briefly wanted to kind of show you, just to give you a flavor of what I did during my PhD. In my research lab at UCL, where I joined, and unfortunately I cannot show you the video because I had to export the presentation. However, what I really wanted to kind of just tell you is that I was very interested in the temporal aspect of cell divisions. So frankly, we spent some time trying to identify methods to, first of all, identify cells in a population, just like as you can see on this slide, and also how do we temporally connect these observations over time, because cells, for whatever reason, they really like to move on the plate, they really like to change their location and their position, but also cells like to divide. And if we want to be able to analyze cells over time, we need to be able to track them or to connect these observations over time, so that we can understand which cell has divided into 2, into 4, into 8, and what led that cell to divide and commit to these divisions at that particular time. Frankly, I found that there was a lot of heterogeneity, and that this process is not as conserved as one would probably think or hope when doing this analysis. And this whole sort of method development and tool development led me to publishing this this paper about the B-drug algorithm, which is something that we've been developing in the lab for a very long time. And I'm very thankful to my advisor for introducing me into this world. So in the next slide, I kind of just wanted to show you that once we got the tracks, and once we got all the information about our cells over time, I really was becoming interested in, okay, well, what do we now do with that information? How can we understand what happens in the cells temporarily? So what I mean by that is, frankly, we were interested in monitoring how cells change over time. We wanted to measure and quantify change over time, and we also wanted to see how this change and change over time differs across these heterogeneous cell lines. So in simpler terms, we wanted to see what makes cells behave differently, even though we shouldn't really expect any difference to start from. So here, during my postdoc, I finished off this fantastic paper that I really enjoyed working on, and it was frankly about using the tracks and the trajectories that we were able to reconstruct from our cells, and being able to temporally measure what is happening in our cells and how they transition through their cell cycle. So for the non-biologists here in the audience, the cell cycle is effectively the process that brings you from one cell division into another one. So turning one cell into 2, turning 2 cells into 4, and exponentially growing your cell population. This project was a lot of fun. I really enjoyed working on it, and I actually enjoyed the fact that it kind of opened doors for me to be able to network and to see the little bit of the world, because I published this sort of project and this sort of research at ICML at the workshop of computational biology, and I was super happy to be awarded the best poster prize at the time. I am not just sharing this image to brag, maybe just a little bit, but what I really wanted to show or kind of emphasize at this slide is that networking is particularly important, especially for early career scientists, and I cannot even say how important it is for the women who want to become leaders in this field, because there is unfortunately not as many of us in this field, and it's very important to get ourselves known in the community. Frankly, the point of this picture and the reason why I'm actually mentioning it here in this slide is that because when I was at ICML last year, what was actually happening in Honolulu in Hawaii, which was a fantastic destination for a conference, I must say, I actually got to know some of my future colleagues from Valence. I met the team there, and I tried to network a lot with them, and I tried to kind of ask what they're working on. And without even knowing that they existed at the time, I kind of, from these conversations and from these in-person interactions, I was really able to see that this sort of company and this sort of team is trying to make a huge leap in the world, and they're working on something that is very close to my heart. So I was really happy to be able to kind of join the team from there. So, okay, now I kind of told you that I've joined Valence, and after my postdoc, I transitioned into the industry. But you might be asking, like, some of you may actually heard the word and the label, the brand of Valence Labs before. Some of you might know Recursion, and some of you in the audience may know neither. So let me just explain what we actually do and what is our role in this kind of ecosystem. You may actually know that there's a lot of companies that are that are acquiring smaller startups or smaller established companies to become a part of their ecosystem. So frankly, the way that Recursion and Valence have done it is that Recursion has acquired Valence, so we're now part of Recursion officially, but Valence kind of stayed as the semi-autonomous AI research engine, which actually has its own mission, vision, hiring system. We have our own headquarters, which are in Montreal, compared to Recursion, which is based in Salt Lake City in Utah in the US. And we're actually the semi-autonomy really shows, because the questions that we're trying to answer are slightly different from Recursion. You may know that Recursion is a drug discovery company, and you may actually know that Recursion is very interested in, and very good at the data generation. And what Valence is good at, and how we complement the Recursion ecosystem, is that we take the quantity and the vast majority of the data that Recursion analyzes and generates, and we actually do a lot of dedicated discovery from the data. So our mission is to industrial scientific discovery to rapidly improve lives, and we do this using AI. And if you're asking, okay, well, everybody does AI these days, how are you guys different from what everybody else is doing? I could probably answer this question by saying that we're actually analyzing and using this data from many different angles and many different perspectives. So we are currently, our teams are spread across many different research units and smaller teams, and we're looking at many different things at the same time. We have a team that is looking at molecular interactions and the chemistry of the molecules that Recursion is interested in. We also have a quantum mechanics and a physical systems team, which are using looking at atomistic simulations and things like that. You'll find more of the details in our hiring page, I'm sure. We also have a biomodality team, which is looking at how we can understand and analyze data across different modalities. And we're also learning from imaging data. And this is where I really come from, is hopefully you're getting the impression that I am an image analyst, and I'm very passionate about imaging and the data that imaging can help us understand. So I just wanted to kind of finish off with a very quick overview, because I've been talking a lot about myself and I've been talking a lot about Valence and Recursion, but I probably haven't given you a good introduction of what is the actual, like, a tangible kind of deliverable effect, or the papers that Recursion and Valence are interested in. So here is one of them. I really kind of wanted to highlight the contribution of Phenom-1. Phenom-1 is this model which Recursion has developed to do self-supervised image representation. And what I mean by that is that because Recursion analyzes and generates a lot of data, they need a good way to being able to represent all of the information that are actually contained in these images which Recursion generates. And Phenom-1 is a fantastic model that can do exactly that. without any supervision, what we do here is that we mask a certain proportion of the image. Here I'm showing you a 5%, used to be a GIF, again, animations not allowed here, but you'll see if you scan the QR code that what Phenom does, what Phenom 1 does in even a higher percentages of masking. And frankly, what this model does is that it learns on itself. It's learning the representations of the images on these mask data using the original image as an answer. And what this gives you is that this model can generate lower dimensional representation, or a mathematical description, if you like, of a particular image, which you can then use instead of the image to describe the key essence of what is going on in these images, what is going on in these cells, how are these cells behaving, and what is representative of their behavior and morphology when you analyze these cells together. So this is just one of the fantastic fantastic papers that has been developed and written in recursion. And frankly, I just wanted to highlight that recursion and Valence are very strong in the impact of their science that they're doing. And frankly, this paper, the Phenom 1 research, which you'll hopefully find if you scan the QR code, you'll find that this paper has been highlighted as a paper at the CVPR 2024, which is one of the major and most prestigious conferences in AI for this year. And it's very soon it's going to get presented there. Next, I kind of wanted to just give a very brief overview of some of the research that Recursion has also been doing, analyzing the vast quantity of the data that they generate. So now, having the representations and the way of extracting the mathematical essence from these images, what Recursion has also done is that they have invested a lot of significant effort into whole genome perturbation screening. And what I mean by that is that they knocked out single gene by single gene, one by one, for the entire genome in the human cell line. And what they've done is that they discovered that some of the knocking out, or the procedure that they actually biologically use, is introducing a little bit of the bias into the system. And they have been able to reconstruct a fantastic map which tells you that where the gene is located on a chromosome is actually quite a complicated. Some of the complications arise because some of the genes that are located nearby may also be affected by this perturbation. And the only way, or the way that Recursion has actually been able to analyze this is because they had this vast quantity of data, and they also had this vast, this massive autoencoder that could actually represent all of the images into a meaningful and a little bit more digestible way of analysis. Again, this paper has been accepted into a very prestigious journal, so I just wanted to kind of show that the Recursion and Valence as well are looking into science which is at the intersection of data acquisition and data analysis using AI, and that also we're interested in the molecular mechanisms in the screens such as CRISPR to be able to understand what these certain molecular scissors do in cells and how we can avoid or be aware of the potential limitations that they have. The third one, actually, and the most recent one, I would say, is some of the research that has been done in our team, in my research unit, in the research unit which I've joined. And this research is focusing mostly on causality. So what I mean by causality is that here our team is trying to learn gene interactions from image data. So now that we understand the limitations of the data acquisition procedure, and now that we can represent these images using Phenom 1, we're actually very interested in learning what sort of pairwise interactions gene may have directly from the image data. So frankly, what we want to do is to discover what sort of pairs of proteins, or frankly, genes, if you think about it in a little bit more of an abstractions, interact in the biological system. And in particular, the biological system that we are using are cell lines. And by interaction, we mean that they somehow are either involved in the same pathway, or there is some sort of a signaling mechanisms there is a signaling mechanism which is somehow making sure that these proteins are interacting on the biomolecular level in the cell. So to do this, we're actually using active learning, and I'm here showing you an image of our team lead, Jason, who has just recently presented this work at iClear at one of the workshops, and his work and the team's work has been, not just selected as a spotlight talk, but also a winner of the Outstanding Poster Award. So I just kind of wanted to briefly show you that there is a lot of mathematical and there is a lot of interdisciplinary angles that we're trying to have a look at. Again, if you'd like to read the paper, and if you're into causality or adaptive learning of genes that can interact in our cell systems, feel free to scan the QR code and you'll find a little bit more. I really just wanted to finish off, with this kind of overview of the research. The reason why I'm not describing a little bit more about my research is because I only joined Valence very recently, so I'm still trying to shape and form what exactly I'm going to be working on. There is a lot of data and there is a lot of research to grasp, so I'm still working on that. But frankly, I wanted to finish off the talk with saying that Valence is a lot about the research and a lot about the AI and the analysis of the data that Recursion generates, but we're also a lot about people. And frankly, that I really don't want it to sound just as a phrase. I feel like a lot of people are saying that they appreciate the interdisciplinarity and that they're trying to form their teams across many different areas. But what I really wanted to finish off with is kind of pointing you towards two of our links again with the QR code if you're interested, because I wanted to point your attention to Polaris. Polaris is a new initiative which is trying to do bring scientists working in the interface of chemistry and biology and provide them with a benchmarking platform. So here Polaris establishes a novel industry certified standard. So if you're interested in doing sort of biological or chemical research, you may find a lot of you may find easy access to a lot of data sets from the world of chemistry and biology, which you may find particularly useful if you're used to training AI models and if you're used to kind of writing papers about these models and if you want to benchmark your model on these data sets. Polaris is still in its beta version. It's not fully public yet, but we're collecting a lot of interest to try, and we're trying to recruit people to try our beta version to get an early access. So if you scan the QR code here, you'll definitely get that. And what I also wanted to mention is that we are currently opening a new office in London. Our headquarters are actually in Canada, in Montreal, but we're also expanding into London to attract a little bit more of the European talent here. And we have a lot of positions across Montreal and across London. So if you're a biologist, a trained biologist like me, or a computational biologist like a wannabe me, please do feel free to scan the QR code or have a look at our website where you'll find a lot more of these hiring open roles and the hiring opportunities at Valence. Thank you very much, and back to Margarita.
Thank you very much, Dr. Christina and Dr. Hani, for your clarity on this fascinating and special subject. Now we will have a roundtable discussion where I introduce the moderator, Dr. Ricardo Nascimento dos Santos, Principal Data Scientist at Sanofi. He will lead this conversation when we will have a more in-depth exploration of the topic. At the end of the session, I will present the questions added to the chat, so I encourage you to input any remaining doubts. Thank you for your collaboration in this webinar, so please, when you can.
Hi everyone, it's a real pleasure to be here. So Ricardo, I have background PhD in biomolecular physics and currently work in Sanofi in France with AI applied to molecular design. So first of all, thank you very much for the invitation and also for this, congratulations for this awesome initiative of Lisbon PH. Thank you, Anna, for the invitation also. And secondly, really nice presentations. Congrats for the nice work present and also the presentations. We are going to wait some questions also from the public to discuss in the end. I separate some questions I think that will be interesting for us to start. One of them, I think that while watching both presentations, I think that it includes both scenarios, is that despite we have all those big changes in the field of AI with the large language models, with ChatGPT, with everything, we see that from my perspective, we are still struggling to have this breakthrough on we're starting that on biosciences, mostly because of, I think that from the point of view of data viability. So that's when we have a lot of textual data, a lot of things from clients, from systems, from purchase still in data. I feel that we have maybe a lack of integration of things. We see, for example, now one of the few breakthroughs, for example, with AlphaFold 3 and on this area that we have a protein data bank invest so much time on this initiative for having enough data that you can leverage that. I'd like to have your opinions of what is the, let's say, the limitations that you see, and mostly is now is data, or still in your experiences are still also some AI development or some new algorithms? What do you think? So maybe Christina can start.
Sure, of course. Yeah, sure. I mean, that's a very good question. I think there are a few limitations that I can think of off the top of my head. First of all, and this is something that I've been definitely exploring at Recursion. When you're generating a lot of data, you gotta find a way to store that amount of data, and that is not always as easy. Storing doesn't just mean to make the data sit somewhere in the corner, but also to make sure that you can effectively access them and that you can effectively kind of make sure that you can manipulate them and access all of this repository when you need it. Now, I feel like with the quantity, like storage is obviously like something that pops to mind to almost everyone when we're talking about the big quantities. But I feel like something I am particularly unhappy about when it comes to big data analysis is that compared to my PhD research, where we kind of had a very limited access to data and we only had a couple of movies to analyze, and it was kind of possible to just walk through the movies, watch them, and kind of like go through the data by eyes. When you start generating data quantities of this sort of scale, it almost becomes untraceable, and it becomes impossible to just spend some afternoon with your cup of tea or coffee in your hand, and it becomes impossible to just go through the data to familiarize yourself with what you're trying to have a look at. And I feel like that is a luxury that a lot of labs, especially academic labs with limited resources, this is something that they can do, because frankly, it's never very enjoyable to look at the data, but sometimes it can be very soothing. It can help you understand that there are some discrepancies in the experiments that you actually never wanted to appear. And it feels like this is something that you can never avoid getting when you're looking at the data at quantities that are petabytes, terabytes, even gigabytes.
I think that's an excellent point, and I would probably add on to the data storage and scale. The kind of the layering limitations on top of that are then accessibility of that data to scientists, and I think taking that even further, also the translating scientific questions into a computer-friendly query then becomes very necessary. If you have this very large data and it's stored maybe in a special kind of data storage system, or maybe in a generic cloud storage like S3 or GCS, you're still going to have to ideally take a scientific question, that's coming from a life sciences perspective, and then figure out how you're translating that. Of course, there are lots of different transformation steps that are being applied to that data, which kind of gets to the traceability that you were mentioning there, Christina. But I think that is a major limitation that we see working with scientists in biopharma that are doing drug discovery, is knowing the kinds of questions that they want to ask, but then not knowing how do I actually ask this question on a massive data set.
Mm-hmm.
Really nice, really nice. Other point, more I think focused to Rani, but also Christina. I have some questions, maybe some focused something that were more broad. One of them is about Pluto. I feel that from what I understand, like it is really nice so state of the art capabilities is really amazing, impressive, and I was saying that it allows data sharing, private data sharing, and also collaboration, and also how that you process that and as automated as possible to have also nice figures for publications. But I see from this and also some potential for being a hub for some of the things that are the main challenges right now for pharmaceutical companies, other companies working in bioscience. That's the digital transformation, getting all those information from so many distinct groups and labs and experimental teams, and then trying to put everything together, centralize, make connect things in a way that they talk the same language. Is that also one of the approach, one of the usages of that? Could you comment a little on that?
Sure. There were a lot of things to unpack there, but what I think is an interesting point that you're raising is we know that we need very clean, structured data, ideally, to be able to just analyze it in aggregate, but especially if we're going to be using AI to analyze that data. One approach that we've taken, especially in the past 5 years at other companies, is making data heavily schematized. And interestingly, when you have a schema that's maybe determined by a computational scientist, where they say we really need all of the samples to have this kind of metadata associated with them. That can work very well for getting very clean data, but practically speaking in the lab, it ends up meaning that you're editing your schema quite often. And so you end up with missing data values because science is very iterative and it's pretty messy, and so we make decisions, we'll run an experiment, we might make decisions about different kinds of data that have to be captured. So interestingly, at Pluto, one of the bets that we've taken is that schemas, we understand are very important and companies may use schemas, but in Pluto itself, everything is designed to be very flexible. And so while you have tabular data like sample annotations or other kind of metadata, you aren't restricted to a particular schema. And because large language models are able to interpret tabular or more flexible, unstructured information, it's still structured in a table, but more flexible table formats. Our hope is that using AI, we'll be able to take that, semi-structured data and draw out the same kinds of scientific questions, or enable us to ask those same kinds of scientific questions. So it's definitely a different kind of approach, and we'll see where it ends up going.
Really interesting, really nice. Now one question more focused for Christina. I think that from the also probably Renid should have a lot of insights on that also, but for cell tracking, for this type of experiment that you were describing, I think that one probable challenge, and that would be interesting for people that are just starting on interpreting those kind of data, is that depending on the image, that the density of cells, the noisy, the type of image formats, type of image quality, or change between drifts between data sets, that would have a very impact probably in the models. If you could comment a little bit on that and give some lesson and learnings maybe.
Absolutely. And I think that's a very good point. And I actually want to bring it up to what Rani mentioned, because I feel like sometimes in biology we're a little bit overly obsessed about the fact that we want to generate clean data. And as a trained biologist, and I'm sure that you will agree as well, I don't think there is anything like clean data in the real world. I mean, no language in the real world is clean per se, by the definition of what we mean by clean as noise-free. There is not a single image that wouldn't have any sort of noise in it. And I feel like it would be very difficult if we just kind of framed our minds into this fact that we need clean data to do interesting science, because this doesn't exist. And frankly, even the cleanest data that you can possibly get, such as when you have an expanding cell line, or when you're trying to track these cells over time, what is happening there is that you find that even in a model where there's absolutely no source of noise, the biology will always become noisy somehow, because biology isn't clean itself. There will always be some heterogeneity, some level of noise. And by noise, I don't just mean like an unclean image, but also noise in the biological processes that we're trying to image, and we're trying to monitor, and we're trying to develop drugs against. So I feel like one of the things of maybe like an advantage of using a big data approach here is that when you have data that are like massively X-fold larger sizes than the noise that you're expecting, in the massive size, the noise is somehow going to vanish. It's going to get canceled out just because you have a lot of data to analyze from. But it doesn't necessarily mean that your models will be noise-free, or that you will work on clean data or clean representations. It just means that the noise will somehow become an inherent part of the model that you're trying to study, whether that's cell lines, whether that's like even patients in clinical trials. There is noise in that data as well. And I feel like kind of making our peace with the fact that our data will never be 100% free and clean will actually bring us to quite a lot of relief, and hopefully to positive results. I don't know if that answers the question, but I really just wanted to kind of bring it up because I feel like noise and the cleanliness of data comes up in discussions quite a lot.
Yeah, really nice point. Maybe also you have any point also, Rina, about from the data that you have seen, the analysis, like the diversity of drifts between data and maybe some low reproducibility depending on the type of data?
Mm-hmm. I think this is where AI will shine, especially with making these larger kinds of aggregate analyses possible, is because it is seeing noise in the system. What AI is really good at is actually being able to make context aware decisions and rationale. And so by identifying the noise and realizing that it's there, it can also reason, and I know I'm kind of anthropomorphizing it a little bit, but it can reason, and it has the ability to draw on its training, which can be very context specific training, to provide answers that are going to be more likely relevant. And so one data set that I've worked with in particular, where actually we applied an ML based approach, but you could, I bet, improve it. Actually, it has subsequently been improved with AI, is looking at pathways, biological pathways that are increased in a cell line when you treat that with some kind of perturbation, specifically small molecules. And when you treat cells in a dish with small molecules, it turns out that inflammatory related pathways almost always, go up. And so that lack of specificity in the response, if you have an AI model that sees that across everything, it can use that as part of its reasoning process to say that, hey, for the scientific question that this scientist is asking me for what biological pathways were altered with my small molecule treatment, maybe I'm going to ignore the inflammatory related ones because that's a non-specific effect. And let me think about what's more unique. So I totally agree with what Christina was saying. There's a lot of noise in the system, and I think where AI is going to shine is in its ability to, selectively ignore some of that noise.
Nice. We have also some questions from the public. How can AI leverage imaging data such as MRA, CT scans, and histopathology images to identify novel drug targets and biomarkers for disease?
Should I go?
Yes.
Okay, sure. So for me, from my perspective, I think there's again, like, the answer, this can be answered from many different angles. I think one of the most kind of obvious angle that I came acquainted with very early in my PhD was that for a lot of time is spent, biologists spend a lot of time trying to do tasks that can definitely be automated. And I feel like AI is a fantastic tool to automate tasks that people do not want to do by hand. Frankly, even if we're just thinking about, this question is asking about histopathology, even if we're just thinking about like counting the number of nuclei in an image, it sounds like such a ridiculous task. Nobody really wants to do it 1, 2, 3, 4, and then you lose count and then you have to go again. And often you're looking at these, slices at these images that have maybe hundreds, maybe thousands of these cells and nuclei in them. So obviously these are tasks that AI can really help you with, because if you had a model that can do the cell counting, you wouldn't have, you could spend your valuable time, either drinking tea, or you could actually do some interesting science instead. So that's one thing. Obviously, like, AI is not just here to count cells and do the mediocre tasks that we don't want to do. AI can actually help us with discovery, and AI can actually, because it's looking at, image data in this particular context, but also any kind of data, it's looking at the data from the point of view that we humans would never be able to do. It kind of complements our understanding. Sometimes what we see from the data, very obviously, when we look at the, I don't know, a cell moving with a, under the microscope, with when we observe the video, the movie, we can see a certain pattern that the computer would never be able to see. For some of us, it's super easy to kind of understand which cell divided into two, and you can really easily connect this into the trajectories. But for a computer, because it's looking at the data very differently, it can be a trivial task for a human, can be a particularly complex task for a computer, and the other way around. For computers, they see patterns that we would never be able to show or see. And this is why it's so important to actually make sure that for any model that we develop, it's going to be we're going to spend some time trying to understand what is the reasoning, what is it using, what sort of pixels, what sort of areas in the image is it using to be able to make that prediction, to be able to make the classification, regression, no matter what we're looking at, so that we can understand how, what the models are looking at, and we can reason whether we can use them in practice or not. Just to give a very kind of brief answer to the question, AI can leverage a lot of research in imaging, because I feel like imaging are images as the product of imaging. These data are particularly data rich, as in we have so many different pixels per image. We have so many different channels per per image. We as humans kind of look at the image, and we kind of are very we're very good at extracting the key essence, but for a computer, they're looking at the images from a very different perspective. And I feel like it's very apparent with these images that is the data quantity that we would not be able to understand by eye. We wouldn't be able to look through all of these images together. But if we had these pattern recognition systems that can tell us what these images are actually all about from a different perspective than what we understand, this is really where we can start using the models to complement our knowledge and hopefully drive us to the new knowledge in the field.
Perfect. Renee, you have some comments on that?
I was looking at some of the other questions that are coming up. No, I think that was really good, Christina. Yeah.
Other point that Renee commented in her presentation that I really sparked a question and some interesting topic in my mind is that the slide that you're describing, how do you identify the exact data point that gives you more information that improves your model, and basically that is, and see that what is the correct question that you can do to improve the information of your model. And over that, I think that it has some relations also with the data Shapley, for example, that allow you to quantify how much does your data value. And on the Pluto implementation, and also Christina, if you your approach on that also, but how is that used in let's say each study is more like you do a new analysis, and on this analysis you identify that you have some part of your exploration space that is not represented well, and then you drives the system towards there. If you could comment a little more on that.
Sure. I think that, yeah, I think you're thinking about it exactly the right way. So there are really two axes, I guess, that I think about when it comes to that solution space that we're exploring in Pluto. One is just the breadth. So how do you add more information, enrich the information in your data set? And like I mentioned, what we're really exploring there are commercially available products and things that we can integrate with, for example, for literature searching, so that if you're seeing some genes coming up, you can maybe go to the literature and see if anybody else has reported that, or likewise go to the data sets themselves and start to ask those aggregate queries of, has this pattern ever been observed maybe in a different, with a different drug treatment or in a different indication? But in terms of the areas where we're really excited about doing our own, proprietary development is when you are doing this process of drug discovery, when you're, creating visualizations, having the AI suggest next analyses to do. And it comes into play in kind of the two different ways that I think were alluding to here, which is either you can have it run all possible combinations, of things, and then make a determination about which of those solutions that it came up with in the solution space actually makes sense to pursue. That's one option. And I think that will be very valuable, and we'll have to benchmark it against other things. But I also think, an area that we're just excited to see really take off is mimicking the scientific method or the workflow that a biologist would typically go through when they're creating their own figures. And so step by step, having the AI make a decision at each point along the way about okay, I did an analysis, I got this result. What would be a next analysis that I would want to pursue? And I think that really lends itself to ultimate explainability in the results that you're getting at. I think where we have to check our own biases here is for a result, a good result that comes out of that, like let's try every solution in the solution space approach versus the more explainable result. Biologists will be more drawn to the one that is more biologist kind of explainable. And I personally am more drawn to that. But I think we'll end up seeing both. And I think, especially with, the papers that are published recently about AI discovered drugs and that are now in clinic and potential success of those drugs. I think that would be an interesting layer to add on to that discussion is, was this drug discovered, or was a particular novel target discovered, or biomarkers, was it discovered by just blasting everything in the solution space, or in a more kind of rational design?
Okay. Christina, any comment on that?
I really wanted to, so I wanted to thank Rani for bringing up this this concept of just saying that, hey, we're experimentalists. We need to make sure that the design of the experiments that we have is well controlled. And this is something that I'm particularly passionate about, because I feel like at the end of the day, whether we're using AI or a pipette or a Western blot or some sort of fancy chemistry machine that I can't even name, at the end of the day, what we're trying to do is science. And for every single scientific experiment that we need to do, we need to have a very good established set of controls that will put our research and the findings that the results that we will generate into some sort of bounds, so that we can understand whether, in cases where we know or we predict what's going to happen, we can get the model, whether that's, again, the Western blot or whether that's AI, whether we can get that model to predict what we know based on our prior knowledge of that system. And if these controls are designed well, and if these controls, the negative ones and the positive ones, can give us some sort of a bounce to what we're trying to do, what we're trying to discover, I think this is already a fantastic step towards making a leap. Because frankly, and again, building up on what Rani has mentioned, it doesn't necessarily mean that to make sure, to be able to leverage the use of an AI model, we have to fully understand what it does. Frankly, this is not even true with drugs, right? There's, a lot of drugs, even off-counter ones, where we kind of suspect what they do, but we really have no idea. But they seem to work, so there is absolutely nothing that should stop us from using them. If we see that the drug is working and it's helping, the patient to recover, or it's, I don't know, helping our hay fever to go down, or we don't have a flu for as long as we would without it, there's absolutely no reason for that drug not to use it, not to be used. And with AI, it's the same thing. Knowing what it does and understanding the mechanism should be key, and it should be a focus of many more labs in the world, hopefully one day. But it doesn't necessarily mean that if we don't fully understand it, we cannot use it. And I feel like the controls or the bounds to the experiments is something that can really push us toward using things, or using models, using agents and algorithms that we do not perfectly understand, but we know that when they're supposed to predict a particular answer, they will get there.
Nice. Also, on the presentation from Christina, when you're showing the methodology for tracking the cells, the single cells, for understanding, you have a method for segmentation. So you segment to identify which are cells. And then you just have a methodology that you develop to track, like where's from the next shot, where's the probable parent shot. I was thinking that you mentioned that also you have some other approach to identify maybe the probability when a cell will divide, and then you also use that to track back to their positions. Does that consider some phenotype, some feature from that you can extract from the image, and or is more based on the time, let's say lifetime of a cell division? And if that considers, the other question is using a super resolution methodology or better images that allow you to maybe differentiate some pixel patterns from different cells would allow to have instant segmentation. Do you think that would be a possible approach?
Okay, so very good question. So the two layers to that question. Let me unwrap that. So the first one is are you using any sort of different features than just the time to connect these observations? I would say Yes and no. A very speculative answer. The yes is because you totally can, and BTrack, the algorithm, actually supports the addition of the features with the belief that these features are meaningful for the tracking. So if you feel like you have features that you want to input into your algorithm, BTrack definitely allows you to do that. There is a lot of software engineers working on the updates and maintenance of the package. So feel free to check that out. The no aspect of that answer is because I haven't used it. I didn't really need to because my segmentations were relatively clear, and the cell model that we were using was relatively easy to track, even though I say relatively because it definitely wasn't. I spent a lot of time doing that. And the second layer to your question was whether using super resolution microscopy would improve the tracking. The answer here would be, I guess so. I haven't tried, so I can't tell you, backed up by results. I can't really tell you the effects of any such experience because I haven't done it. But I know that BTRAC has been used across many different microscopy modalities. So I would say that it definitely is possible, and you can you don't have to have a specific cell line that I had and a specific microscopy setup that I had to be able to do the tracking. You can absolutely extend it to any sort of systems that you would want to do.
Nice. Another question focusing for Rennie. The Pluto could now could use ingest some data, for example, from Christina, some data with single cell trackings, and like if you could discuss a little more about how it deals with unstructured data or other types of data like images and different data, and also which are the most frequent data sets today if we just have let's say a summary of the types of data that now in Pluto are more deposited, let's say.
Yeah, so I love the image analysis side of things, and I would love to collaborate with you on that, Christina, because that's something that we really don't do in Pluto. But when I got my start as a researcher, I was doing live imaging on a confocal with zebrafish, looking at the central nervous system, and I miss those days so much. So, and that was way basic compared to what you're talking about. So, no, in Pluto, really what we're mainly focused on would be omics data sets, and so those are those molecular high throughput data sets. They can be sequencing based like RNA-seq or ChIP-seq, but they don't have to be. They can also be using a mass spec in the case of proteomics or metabolomics. And the reason for that really is because multiomics, and I see one question in the comments there about multiomics data analysis, but multiomics is just kind of a hot topic and an area where I've been really excited because I think it follows the sort of central dogma of biology in a really interesting way. And then I think I would see imaging as probably this final phenotype. So maybe, post the central dogma. And so that's where, we kind of stop. So the most common kinds of data that are analyzed in Pluto today are gene expression data, epigenetics data. And then when you start to combine those, you can ask, what is the epigenetic profile here? So what does DNA occupancy look like? How open or closed is the chromatin here? And then how does that correlate with gene expression? So you can start looking at all of those pieces, DNA, RNA, and then protein expression even, and start to ask that kind of biological question. So that's where we're at today. In terms of, like I said, loosely structured data, all of those assay types follow the same kind of format that I highlighted in my talk. And so when you have that basic context and you provide AI with the context around this is gene expression data or this is measuring DNA occupancy, and these AI models clearly understand that quote unquote central dogma of biology, they can start to draw inference in that way.
Nice. Perfect.
So more imaging data needed. I would love to see more algorithms developed there. And I imagine, or I've heard from researchers in the lab that the usability of some of the imaging software is lacking. So I'd love to be able to make an impact in that someday.
On comment on that, now with, for example, with cryo-EM, there's so new technology that you have so new amount of new data that is coming up. It's really, really amazing. One other question from the audience is, what is the most effective computational approach for identifying and quantifying bias in CRISPR screening data, and how do they inform subsequent correction strategies?
I'm going to take that question because I kind of feel like it's for me. Cool. So not to bore the audience and give like a 30-second kind of answer to that question, which is a fantastic one. By the way, thank you so much for asking. I think the answer to that would be to identify the bias raised from the CRISPR datasets. What is important is to be able to perturb every gene individually, which is something we've done. Our team has done at Recursion. And so what they've done is that they've introduced a perturbation to a single gene, and they actually found that genes that are spatially very close to each other, but not functionally. So what I mean, they're located close to a chromosome, but they have nothing to do with each other. They have completely different functions. They have found that even though the functions are very unrelated, the cells, the resulting phenotype under the microscope, they will look similar. And the only reason why they kind of the team reasoned that this is occurring because there is actually a loss of a lot of DNA material that is causing them to look the same, even though the function is unperturbed. So this paper has been out there for a very long time on bioRxiv, and is hopefully getting there as a formal publication in the next couple of weeks. So feel free to dive deeper into the preprints for a more kind of clarified way of understanding the science behind it. But frankly, to be able to do this, first of all, you need a lot of data quantities, as the topic of today's discussion is revolving around. And also, what you need is a good way of being able to represent these image data. So once you take the images of all of the perturbations, you need some representation tools like Phenom1, but not only solely and exclusively that. You need some representation tools so that you can compare these representations together mathematically, quantitatively, whatever smart word you want to attach here, and so that you are able to quantify the bias and similarity of the genes, even though they're not functionally related. So hopefully that answers your questions, but the preprint is out there. Feel free to have a look.
Nice. Other question I think focused for Renee is what collaborative opportunities does Pluto Bio offers to enhance AI-driven research in the pharmaceutical and biotechnology industries?
Interesting that it's collaborative opportunities, so, and then enhancing AI-driven. So I guess I'll caveat a couple of things. So first of all, a lot of what we do with Pluto, especially around user experience and just this whole research experience, does not have to be enabled by AI and isn't currently. So a lot of what Pluto is seeking to do is just make it possible for interdisciplinary scientists to be able to communicate their research and have a common medium, or canvas, we call it, for drawing out those different scientific questions and workflows. So I think collaboration, can happen with a better user experience and a lot of other aspects of the platform. And then I think for enhancing AI-driven, let me see if I understand the question. Enhancing AI-driven research. I think I don't want to sound like a broken record, but I think really the biggest opportunity that we see here for the AI-driven research that we work with is in the multi-omics space. And so there are a couple of different ways by giving a platform to different kinds of scientists to be able to input their data in a friendly way that doesn't necessarily have to be overly schematized or structured. You're now creating an environment where all of that data lives, and then you can start to ask more advanced questions like multi-omics. And so typically the approaches to multi-omics have historically been, we'll take a really large data set, train an AI model on some known multi-omics data that was maybe used to identify a novel target or biomarkers of disease. And then if I have a similar kind of multi-omics data set that I've generated, I can run that through the model. This requires a really large data set though. And so really where I see Pluto heading for this AI-enabled research and collaboration is in helping researchers that have much smaller data sets, either through that emulating a biologist's thought process approach, like I mentioned, or by pulling in other data sets to do comparison against. And that is also a very collaborative process, using other people's data that they've published, hopefully connecting with those researchers also and being able to share the results more from kind of a community, research community perspective there. So I hope that got to the root of the question that was being asked here.
Perfect. I have also other question here. One for Christina. It's when you mentioned about the proximity bias and whole genome perturbation screening. So the idea, if you could comment more from the student, it would allow to also based not only to measure the effect of the physical distance uh, of the positions of in genome, but also to have maybe some map of knowledge graph of how some knockout of some gene could affect the entire system, or which let's say have some idea of interaction force basically between those
Absolutely,
genes.
absolutely. I think you're asking a very good question here, but frankly, I think the problem with the proximity bias is that what you just described, proximity bias, is an off-target effect of that. Because if you want to study the interactions, you want to make sure that when you're studying the interactions and the pairwise kind of contributions of one gene to another and how they're related in pathways, you want to be sure that your signal is clean. And again, we're coming back to the word of clean, non-noisy data, and this is something that is very difficult to generate. But frankly, what you do not want to have is analyze the signal that is arising from a perturbation of different genes than the one that you're interested in. And frankly, proximity bias is exactly about that. So proximity bias is telling you about, this phenomenon that when you look at the whole genome scale, there are genes interacting together, or they appear to interact together, even though they're functionally dissimilar. And the only reason for that is because the perturbation actually perturbs, or it actually kind of modifies or affects the expression of the genes that are nearby on where you're interested in. So frankly, proximity bias is something you want to get rid of, not necessarily something that you would want to use, but you described the kind of the mission and vision of why we're doing these screens in the first place really well, because exactly we want to we want to generate the map, we want to understand the interactions of these genes, and we want to be able to understand which genes interact with each other, which genes are interacting with each other but in a negative way, which genes are inhibitory one to another. And to be able to generate that, we actually have to do a large screens, and we have to gather a lot of data which are not proximally biased if possible.
Thank you. Nice.
In this manner, I bring the third and last webinar to a close. I hope it has been enjoyable for the audience. To our speakers, I hope you have enjoyed collaborating with us as much as we have enjoyed listening to your expertise on this topic. I'd like to draw your attention to the QR code present on the template displayed on the screen. By scanning it, you will have access to the satisfaction survey for the session. I appreciate the response from all participants so we can improve our service. Just like this one, the previous two webinars were broadcasted on YouTube, and both sessions can be found on our YouTube channel, ListenPH. If you missed the chance to deepen your knowledge on the topics cell therapies for cancer or frontiers in aging, principles of longevity, this will be the perfect opportunity to watch these sessions. Thank you once again for your presence.